Okay, so this is the first vision lecture, and in this lecture I'm going to be talking about the gross anatomy of the eye and the organization of the retina. So when thinking about what the eye is doing, I divided its jobs into two broad groups. The first is to focus light, and the second is to detect light. So focusing light primarily uses the cornea and the lens, and then detecting the light uses the retina. So first we're going to look at focusing the light. Why do you need to focus light? Well, when a light source, like this happy sun here, shines its light down onto an object, this flower, the light diverges in all directions. It bounces off the flower, it diverges in all directions. A few of these rays of light might enter my eye, but they're still diverging. So it's the job of the cornea and the lens to bend that light, to refract that light back onto a single point on the retina. So now this little spot on the retina gets all the light from a single spot on the object that we're looking at. And when this happens over the whole retina, we get an image of the whole object. So what's the anatomy that enables this focusing? So I split the anatomy slides into two slides. The first, I'm going to cover what I call the transparent bits, the colorless, see-through pieces that the light travels through. So the light first, when entering the eye, goes through the cornea. So it's the tissue on the surface of the eye that begins the refraction of the light. It bends the light by about 40 diopters. That's a measure of refraction. Light then passes into the aqueous humor. This is a watery fluid like the serum in your blood, and it supplies nutrients to both the cornea and the lens. And of note, glaucoma, a very common eye problem, is caused when the aqueous humor doesn't drain out properly. So the aqueous humor is continuously being replenished, and when it doesn't drain properly, pressure builds up. You get high intraocular pressure that pushes on blood vessels. It makes it so the retina doesn't get enough blood, and ultimately neurons are damaged. Okay, so back to the light path. Light paths through the cornea, through the aqueous humor. It then goes through the lens. So lens is this flexible biconvex tissue. Looks a lot like a lens that you'd find in a camera. And this refracts the light some more. It kind of finishes the focusing of the light so that the light then focuses on the retina. And it does it with about 20 diopters of power. And I put the diopter value in here because it's maybe a little interesting, because when we talk about focusing of light, we usually think of the lens, but really 2/3 of the focusing power comes from the cornea, and it's just that last third that is provided by the lens. So the light passes through the lens, it then goes through the vitreous humor. Vitreous humor actually makes up the bulk of your eye. It's this gelatinous mass somewhere between a liquid and a solid. And not a whole lot going on here, but it does have phagocytotic cells that crawl around and remove debris, the little floaters that you might see in your eye. So for your first summary question, answer, why do we need light to be refracted in the eye? Why do we need the cornea and the lens to bend light? Okay, now on to the more dynamic bits of the anatomy of the eye. These are parts that move and do things. So first off, in front of the lens, behind the cornea, we have the iris, and this controls the size of the pupil. So when it constricts, the pupil constricts, and you have less light entering the eye. This would be like if it's very bright out and you don't want to damage your retina with too much light. And then if it's dark out, you could relax the iris, let in a lot of light, be able to see things better. Moving on, you have the ciliary muscles and the zonule fibers, which are attached to the lens. And these are important for controlling the shape of the lens, allowing for accommodation, which we'll talk about in the next slide. And finally, I've already mentioned it, this neural structure at the back of the eye, the retina, which detects light, it processes those light signals, and then it sends that off to the brain through the optic nerve. So accommodation, what is it? Well, light must always be focused on the retina. We talked about that in like slide 2. The problem is objects aren't always the same distance away. So I found this simulation of accommodation website that I'll pull up here. So basically, here an object, light rays are diverging, the lens focuses it on the retina. So everything's good. You have an image forming on the retina. But if the object moves away, the image is now forming in front of the retina. If some object gets too close, now the image is not forming on the retina. So we need to have a flexible lens, a lens that can squish and get really fat, or it can pull and get really long and thin to allow its refractive power to change based on how far away the object is. So that's why we need accommodation. So a rounder lens has stronger refractive power and can focus on closer objects, whereas a flatter lens is for focusing on more far away things that don't need the light rays bent as much. And so the lens can change its shape to adjust the focal length of the entire eye. And it does that, as I already mentioned, with the zonule fibers and the ciliary muscles. So how that works is the zonule fibers are always pulling on the lens. They're pulling it flat. The ciliary muscles, when they contract, they actually release tension from the zonule fibers and allow the lens to return to its more natural round shape. So you relax your ciliary muscles when you want to look at something that's far away, and you contract the ciliary muscles when you want to look at something that's close up. But this goes wrong for a lot of people. So I am nearsighted, I am myopic, and the problem with myopia or hyperopia, nearsightedness or farsightedness, is the image plane is not occurring on the retina. The light rays are not converging where they should converge on the retina. So here's the 3 different possibilities. You have emetropia, which is just normal. The light rays are converging on the retina. Myopia, here light rays are converging in front of the retina. So this is very common. It can be caused by either a cornea that is too curved or an eyeball that's too long. Most of us, our eyes just don't stop growing when they're supposed to stop growing, and your retinas just move beyond the image plane. And so you can relax your ciliary muscles to try to see further, but there's only so much they can do. It's outside the range that they can relax. You can never get your lens quite flat enough to see what you're trying to look at far away. And then there's hyperopia, which is just kind of the opposite of myopia, where you don't have enough refraction of the eyeball, or the eyeball is too short, didn't grow long enough, and now the image plane would be occurring beyond the retina. So these are very common. And then a third, or a fourth, I don't know, another problem that we have with image planes is presbyopia. And this is when the eye's ability to accommodate is reduced, and it makes near vision tasks difficult or impossible. And so this is just really kind of a natural thing that happens with age, where the lens, which is its elasticity, its ability to deform, is necessary for accommodation. That just goes away with age. So your eye shape might be normal, you never needed glasses, but then as you get old, you might need to start wearing glasses. Or if you're farsighted like me, as you get old, you might need to wear bifocals because you can't focus either far or near. You can't adjust. So here this graph shows that young people, their eyes can change, the lens can change a lot. It can change up to 13 diopters. Then as you get old, it goes down to basically 0. It has a set dioptic power and can't change very much. So this brings me to the second question for you to answer in your summaries, and that is, why does the lens need to be flexible? Moving on to the retina, which we're going to spend a lot more time on in this lecture and next 2 lectures. So some features of the retina that you can see if you look through the front of the eye with a visual ophthalmoscope. The first is the optic disc. This is where all the blood vessels enter and leave the eye, also where all the axons of the ganglion cells leave the eye. And since it's full of blood vessels and axons, there's no photoreceptors there, so it gives you your blind spot. It's a spot in your retina that can't see, and you've probably seen this if you do one of those tests where you look at a spot and then close one eye and a second spot goes away. But usually the blind spot's not a problem because our other eye covers for it. So then this other spot over here on the eye, this is the macula, and then in the center of the macula is the fovea, and these are areas of dense cone photoreceptors that support high acuity vision. And so we actively alter our gaze, we move our eyes around so that whatever we're looking at is focused on the fovea. And that's where most of our visual processing occurs. And Dr. Schmidt put a picture of her retina on the slide, so I figured I'd throw a picture of my retina. This isn't through a visual ophthalmoscope. It's actually a picture of all my blood vessels in the retina, but you can decide who has the prettier retina. So how is the retina organized? Well, it's a little weird. This is a cross-section of the retina, and you see these different layers of cells, and the light actually enters. So this would be like light coming through the vitreous humor, and then it enters the retina and passes through all the layers of cells until it reaches the back of the retina, where the photoreceptors are, which is just strange that it passes through blood vessels and neurons and gets all the way here to the back. And then those signals are actually passed through the retina back to the front, the ganglion cells, and the ganglion cells' optic nerves head out through the optic disc to the brain. So we're going to look at this in a little more detail. And it looks like a bit of a mess, but the retina is a nicely layered structure. So there's 3 layers of cells. This layer out here, the outermost layer is the outer nuclear layer. Then the next layer is the inner nuclear layer. And then finally, the ganglion cell layer. And between each of these layers of cells are layers of synapses where all these cells connect to each other. So these photoreceptors connect to the bipolar cells in the outer plexiform layer, and then the bipolar cells connect to the ganglion cells in the inner plexiform layer. And you can categorize the flow of information through the retina into 2 groups. The first is the vertical information flow. And this is the excitatory direct pathway. So excitatory is in releasing glutamate. And this is simply photoreceptors detect light. They release glutamate onto bipolar cells get excited, and then the bipolar cells release glutamate onto the ganglion cells. And I put in here the different types of the cells because along this pathway, you get more and more cell types. So initially you have the rods and the cones, and you have a couple different cone types. We have 3 cone types if we're trichromatic, but you only have a couple cell types. Then you move on to the bipolar cells, and you have several types. You have on types that respond only to light turning on. And you have cells that respond to light turning off. And then within this, you have multiple types, some that are more fast responding, slow responding. And then the ganglion cell types, you have even more diversity. You have cells that respond to light on, off, and even more specific features of the visual environment. So you just kind of get this more and more diversity. And this is partly caused by the horizontal information flow. So we just talked about the vertical information flow, but at each of these synaptic layers, there are cells that stretch laterally and inhibit. So they inhibit with GABA or glycine. So at this first synaptic layer, you have the horizontal cell, and it inhibits the photoreceptor to bipolar cell synapse. And I put in here luminous and color because there's, in humans, 2 types of horizontal cells. One that mostly just responds to light level, and then one that is actually changes its response dependent on color. So we can get some diversity at this first synapse. And then at the next synaptic layer, the interplexiform layer, you have amacrine cells, which extend laterally. And there's a whole bunch of types of these. And they inhibit the bipolar cell to retinal ganglion cell synapse. And some of them are small and do very local inhibition, and some of them are really big and extend very long distances, and can carry information from far away parts of the retina. And then the last thing I wanted to talk about for this lecture is graded potentials versus action potentials. So most of your brain uses action potentials, but actually most of the retina uses graded potentials. And there's a good reason for it, because graded potentials have high fidelity. They're an analog signal. So if you remember, this is from one of Dr. Schmidt's earlier lectures, graded potentials, you can have, a small response, a bigger response, a bigger response, a bigger response, you can, or anything in between to increasing stimuli. Whereas action potentials, it's binary. You might not have any response, and then you jump to one spike. There's nothing between 0 and 1 spikes. So graded potentials have high fidelity. And so it's used by most of the retina. It's used by the photoreceptors, everything out here, the bipolar cells, and most of the amacrine cells. But there's a reason for action potentials, and that's because action potentials allow for long distance signaling. So once you convert that graded potential to a digital signal, to a spike, you can send that a long ways and not lose that information. So really only 2 types of cells use these spikes in the human eye, and those are some amacrine cells, the really big ones that have to stretch out a long ways across the retina, and then retinal ganglion cells, because their axons leave the eye and travel all the way into the brain, so it's a very long distance, so they use action potentials. So for the final question for your summary, I want you to tell me which cell types in the retina fire action potentials.